welcome back to the lectures on animal physiology in the NPTEL. So, we are in section 5 with the nervous system. So, today is the lecture 7th. Okay. So, as of now in the nervous system, if I just have to have a recap, we have started with the structure of neurons. We have talked about the supporting cells, the glial cells and their structure and function. We talked about myelination, and then we talked about the overall architecture of the nervous system, the central nervous system, peripheral nervous system, and then we talked about the reflex circuits, talked about the higher brain functions like memory and learning, and we talked about the two learning models, long-term potentiation, long-term depression, one of which is based on Hebbian learning model, and then regarding Alzheimer's disease and dementia. So, today what we will start is we will talk about few other diseases of the central nervous system, which has helped us to learn a whole lot about the way our nervous system functions. It is very interesting that most of these informations what has been gathered in last 100 years are mostly have evolved from clinical 1940s, when this part of the hippocampus or the hippocampus was removed from the epilepsy patient, that leads to the loss of memory, like this person never acquired any further memory, and that set the tone to understand hippocampus and the energy storage mechanisms followed in the hippocampus for next almost now 70 years, and it is still continuing, and it is continuing at the molecular level with enormous understanding and intense applications of electrophysiological techniques into it. Okay. Whereas, that was the beginning of long-term potentiation, which was in 1970s, followed by 1980s, when the long-term depression model, it was given by Masao Ito, and afterward people like Taras Sajnowski in Salk Institute and few other people who have done significant of work, and in India, National Center for Biological Sciences, Professor Sumantra Chatterjee, he is doing a lot of work on amygdala, and some really groundbreaking works have been done all over the world to understand how our emotional memories are being stored and are being carried forward. And in that process, while explaining the long-term depression, I kind of highlighted the fact that there are neurons whose threshold to respond reduces because of long-term depression, just as opposite to the long-term potentiation phenomena whose threshold increases. Here the neuron thresholds decreases because most likely because of it has a different kind of neurotransmitter profile, and it's on its membrane, the channels densities may be totally different as compared to the neurons in the other regions of the brain. Okay. So, with this, let us talk about one of few other diseases. We will be talking briefly about epilepsy, again relevant to hippocampus. We will talk about Parkinson, Parkinson's disease, and therapies. And then we will be talking about spinal cord damage and futuristic therapies. These are the aspects what we will be dealing in these three topics. What we will try to do in this lecture or in the subsequent lecture, we will wind up these, or at least my objective is that you people should be aware about the state of art across the world where we are. That will help you to kind of go through any kind of research papers or the findings, and you should be able to see that how far we have progressed. Okay. Coming to epilepsy, so the first one to deal with. Okay. What really is epilepsy? In the last class, I explained you that epileptic patients all of a sudden gets about. It's kind of about when they, the first symptoms, if I don't know how many of you have seen an epileptic patient, they like move like this, likewise, and then they faint down, like the whole body starts shivering, and then they fell down, and you will see sometime from their mouth, some kind of saliva started coming in, and they are not aware, and just after some time they are all fine. They just got up, they feel very tired, but then they have no idea what happened few minutes back, because that part of the event at that time t is totally washed out. They have no idea. So, how that happens? It is something all of a sudden. Think of a crossing on a road. Okay. All of a sudden the vehicles start coming from all sides and they collide. There is a huge bang, and then everything becomes quiet, and obviously you remove the debris, and then again the traffic starts moving. So, it is something very similar to that what happens in epilepsy. There is a hyperactivity in the hippocampal region of the brain. So, it is something like this. If this is the structure of the brain, and here you have the hippocampal region. So, all of a sudden there is an uncontrolled hyperactivity here. So, what happens immediately is, due to this hyperactivity, sudden hyperactivity, the impulses are being transmitted all over the place, all the connected circuits, and as I told you that all the circuits are connected. All of a sudden all the coordinations of the body is kind of lost. You are just have no proper signal, as if there is a collision on a four-way crossing, and that collision could be because of, what are the possible hypotheses out here? Could be because of all of a sudden, if you break up the problem at the cellular level, all of a sudden there is excess secretion of neurotransmitters. So, sorry for the glitch, there were some errors in the systems. Okay. So, there may be excess secretion of neurotransmitters, uncontrolled secretion of neurotransmitters, and most of these neurotransmitters which are involved or which is believed to be involved is glutamate, as I have already mentioned that this is basically most of the hippocampal region are consisting of glutamatergic neuron. Okay. And this glutamatergic neuron all of a sudden secretes a lot of glutamate. So, the problem with glutamate is that glutamate may lead to excitotoxicity. This is another problem with the glutamate, because it has to be removed from it. So, there is a baseline up to which a body or the brain can tolerate, but beyond that it becomes toxic, and that leads to further complications. Okay. So, one of them may be glutamate, or some way or other there is another aspect to it. So, while talking about the hippocampal region, I told you that within the hippocampal region you have glutamatergic, GABAergic, GABA secreting neuron, glutamate secreting neuron, and there is a balance between the numbers. Okay, there is a proportion in the numbers. There are certain numbers. So maybe there are brains which have developmental issues, or developed in a totally different way. Their number of the GABA may be much lesser. So GABA, which is an inhibitory neuron, which allows some of the chloride and all this kind of ions to move through, that may be less. So that may lead to an hyperactivity and excess activity of the glutamatergic neurons. Okay. So whatsoever be the situation, it must have to deal with the neurotransmitters and the ion channels, or there may be some mutated ion channels which are formed, the one which are conducting all the, so maybe an ion channel problem. Okay. So at the level, if you look at it from very simple chemistry perspective or biochemical perspective, it will boil down to either neurotransmitters or ion channels. And now if you go with a reference to the first chapter or the second chapter what we covered in the membrane physiology, so they are all membrane related phenomena, and that is why there is intense amount of funds across the world in best of the best places like National Institute of Health or in UK. There is enormous grant which are given to understand the membrane channels structure and function, because these membrane channels are the one which ensures what is entering inside the cell and what is going out, and that is the hot target for all the drugs to bind and carry out all the downstream functioning or executing their pharmacological effects. Okay. So with this brief idea about epilepsy, we will move on to the next pathological condition, which is Parkinson disease and its therapies. So Parkinson's disease and its therapies. Okay. What exactly is Parkinson disease? So in order to understand Parkinson disease, we have to rebuild the circuit. But before I build the circuit, let me tell you the problems a Parkinson's patient faces. A Parkinson patient cannot move his or her, the motor controls are compromised. In other words, they cannot move their hands or their legs or several other parts of the body do not move. What exactly that means whenever we see a patient of Parkinson? So now if you recollect the circuit of neuromuscular junction, so what is happening? There is a sensory input from your muscle which goes all the way to the spinal cord, and from there part of it goes to the brain. From the brain, if it is not a reflex circuit, of course, there are two options. Okay. Either it is a reflex circuit, it goes to the brain, it goes to the spinal cord, from the spinal cord it comes back. Okay. So just for a recap, so this is the brain, this is the spinal cord, so and this is the target tissue, and these are the sensory neurons, and these are going cell body out here. And if it is a reflex circuit, then the motor neuron which are sitting on the spinal cord, they come back and tells you. So this is the classic reflex circuit what we have already talked about. Now there is a second circuit which needs higher motor controls. So the motor neurons can be divided into two parts. Okay. So one which I have already mentioned here, these are called spinal cord motor neuron. Spinal cord motor neuron. Their cell bodies are sitting on the spinal cord, and their processes goes out to all the target tissues. These are sometimes also called SMN, also spinal cord motor neuron. Okay. So they are all sitting all along this ventral root. And if you remember when I talked to you about the dorsal and the ventral root, just again for a recap. So the center is the ventral root out here, and on on both sides is the dorsal root, where the sensory root or the ascending pathway is being there. So, this is the ascending or the dorsal root. Okay. It is ascending because I told you all the informations are carried like this to the brain on this, and this is motor, where the signals are being brought back from the brain or directly from the spinal cord like this. Okay. So, the ascending and the descending pathways. Coming back to where I was. So, there are other circuits where basically a signal from, say for example, here a sensory signal from, let me draw another situation. Okay. This is a sensory ending which is moving all the way. Here is the ganglion. From here along the ascending pathway, this signal is moving to the brain. Say for example, here it reaches the motor cortex. And just for your again recapitulating some of the myelination thing. So, these neurons which are sitting partly outside the central nervous system are myelinated with Schwann cells, and the one which is the part of it which is sitting inside the spinal cord is myelinated with the oligodendrocytes. Now, from the motor cortex, it synapses on another set of motor neurons. Say for example, it is in the motor cortex. These neurons are called higher motor neurons. Higher motor neurons. These higher motor neurons are present in the motor cortex of the brain. Motor cortex is the region of the brain which regulates the higher motor control or coordinate the higher motor control. How it does so? One second. Higher motor controls. So, the way it works is from the brain, from the region of motor cortex, which are sitting somewhere out here, series of thousands of motor neurons, they send the signal to the lower motor neurons. So, here in the blue you have the lower motor neuron on the ventral cord, okay, and that is where they are synapsing, or some motor neuron sitting here, they are synapsing, and these signals are then being sent to the target tissue, and here you have the target tissue, right. So, the way signal is moving is, here is a sensory input going all the way to the brain, either via interneuron or directly by synapsing on the higher motor neuron. This signal is being transmitted, signal moves like this all the way to the brain, and from here the signal starts coming down. Likewise, it reaches the target tissue. So, in that process what happens, signal reaches here. So, the higher motor neuron commands, and this command is being executed by these neurons. Imagine a situation when selectively some of these higher motor neurons which are present here starts dying. Just putting it higher MN, they start dying, and if this happens, next thing what you know will happen is this: the signal or the electrical impulses which are supposed to be transmitted along this line will be compromised, they won't be sent. So, under that situation, option is that this person is no more receiving the higher motor inputs, and this may lead to unable for this person to move his hand or her legs, or all other body parts. This is the classic symptom of Parkinson disease, and the series of neurons which starts dying at the motor cortex are especially in a region of the brain called substantia nigra. This is the region of the motor cortex, and the motor cortex which is responsible for coordinating the motor control. The specific set of neurons which starts dying are called dopaminergic neuron. Now, if you dopaminergic neurons, now if you people go back and see the classification of neurotransmitters, you will see there is a series, one class of neurons or neurotransmitters, where serotonin, dopamine, all the amines, it is that class of neuron. There are a population of neurons whose, so it is like this. It is a, so these higher motor neurons. So on receiving input from the sensory neurons, secretes dopamine. So this dopamine is synthesized here. These cells are specialized to synthesize dopamine, and this dopamine act as the neurotransmitter entities. These dopamines travel all along here, or maybe synthesized out here, and these one forms synapse on the lower motor neurons out here through dopamine. It is the dopamine which is transmitted from here. So now there is no more dopamine. Dopaminergic neurons are no more there. They starts dying, and under that situation, it is not getting any further signal. This is the classic case of Parkinson disease. What are the difference between a Parkinson patient and an Alzheimer's patient? So, AD, Alzheimer's disease, and PD, Parkinson disease. In Alzheimer's disease, the majorly affected region is the hippocampus, and in hippocampus, whereas in the case of Parkinson disease, it is substantia nigra of motor cortex. First difference. The second difference is it leads to dementia or memory loss, conscious memory loss, dementia or memory loss, whereas in the case of Parkinson, no memory loss. Whereas in the case of Alzheimer's disease, motor control is intact. There is no problem in the motor control. Motor control is compromised. These are the very fundamental difference of these two different kind of diseases which happens in the brain. And if you look at it, what I wish to highlight here is that whenever you think of a brain, think there is a mass of neuron all over the place, but if you just think a little bit in depth, you think of it, a disease which is happening here, and if, for example, this is the region of the motor cortex, okay, which is all the substantia nigra and all other regions of the brain. Or say, for example, let's talk about a disease in amygdala, okay, and this is your hippocampus. So it is the same mass of neurons, but the way they succumb to disease is totally different. So, for example, if this one, it has two diseases we have discussed, Alzheimer's disease and epilepsy. It is a totally different kind of way it works, okay. And talking about motor cortex, we talked about Parkinson disease, and talking about amygdala, there are depression and fear psychosis. So, if you look at these neurons, what is there a central way we can put them? Of course, they all have one common thing, they are all electrically active, but they have different kind of neurotransmitter profiles. At the molecular level, at the level of their membranes, they are totally different. The substantia nigra neurons, they secrete dopamine, which is being transmitted to the lower motor neurons, and then the signals are being executed. In the case of hippocampus, you have the glutamatergic neuron. They secrete glutamate, and this glutamate is being then helps in communicating with other neurons. You have the amygdala. They have a lot of serotoninergic neuron. They secrete serotonin. So, it is the same brain, but the fate of different neurons are totally different. And what is the most fundamental question, one of the very, very fundamental question in neural development, how the nervous system developed, is how a neuron decides that it will become glutamatergic, that it will secrete glutamate, or it will become dopaminergic, how it will secrete dopamine, how it decides that it will become cholinergic, that it will secrete acetylcholine, how it decides it becomes GABAergic, it secretes GABA, or serotoninergic, because this question has to be answered in the years to come. Why is it so? Because now by this time it is clear to you people that all these different zones are dictated by different set of neurons. So, now when we are ushering into the era of regenerative medicine and tissue engineering, where people are pulling out different kind of stem cells, which has the potential to become any fate. So, for example, from the brain you pick up a set of stem cells and you wanted to look for a therapy. What will you do? So, it is something like this. Say for example, this is the brain, and you know a part of the brain or any part of the tissue, okay, which could be transformed into, say for example, some stem cells are coming out, okay. Let us call them as neural stem cells. So, these cells will form neurons, okay. If they are given the proper conditions, they will form neurons. And now, what will decide that these neurons could become? So, do you have a control mechanism, or do we as human race have a control mechanism to tell that okay, under these conditions, these will become say cholinergic, okay. Under these conditions, they will become say glutamatergic, or under these conditions, if I grow them in under this condition in a dish. So, what you do essentially is that you take a stem cell population like this, okay. Then next thing, so these are the stem cell population, and next thing you do, you grow these stem cells, you divide, make these stem cells to divide, okay. Stem cell division. The next thing you do, so the dream therapies are like this, okay. Next thing you do, you pull out a specific population out of it like this, okay, and then you differentiate them. Differentiating them means you decide what kind of fate they will have. Will they? The first question is, could I make from a stem cell population a motor neuron, or could I make a pyramidal neuron of the hippocampus? Could I make a neuron of substantia nigra kind? Say for example, I say for example, here is a patient. So, in future, in a distant future, this is where our therapies will be. So, if say for example, a person suffers from say Parkinson disease. So, this is the zone where the neurons are dying. Now, what will happen? Distant future is that okay, I know these are, he has this kind of stem cells, neural stem cell in neural stem cells, okay, NSCs. These neural stem cells will be divided in the lab, and then they will be differentiated to form dopaminergic neuron, the one which secretes dopaminergic neuron, and then these dopaminergic neurons will be transplanted back to that part of the brain where they are dying out, transplantation. Okay. With the hope that these dopaminergic neuron, newly transplanted dopaminergic neuron will form the same connectivity. So, that is another whole different ball game. This is at least what is being dreamt of as the futuristic therapies for mankind, if one day, and this is going to happen. So, for the generations now, the challenge lies here. We all now have well characterized stem cells from different parts of the body. These cells, it could be even a skin cells, you could have a skin cells and you can make neurons from them. Okay. From skin cells you are making neurons. Now, these neurons have to be transformed into, say, a motor neuron. Could we do that? Or I say I want to make from these skin cells, say, oligodendrocyte. Could I make that? Or when I am talking about, could I make, say, for example, a cardiac myocyte, since we have already studied about it, cardiac myocytes. Could I make that? Because skin cells are most easily accessible, skin stem cells, okay, or maybe some skin cells. Okay. Or could I make, say, dopaminergic motor neuron? I just put DOP as dopaminergic. Okay. Or could I make hippocampal neuron? And even if I could make them, could I decide that this will become cholinergic or this will become dopaminergic? So, these are the challenges for next few generations, that how far we can control the fate of a cell. And then comes the next challenging part, what I was trying to highlight in the previous slide, that transplanting it back to the brain or specific different parts wherever it is needed and ensure that they form the circuits. And this will be, these kind of experiment in the future will surely help us to look at neuroscience from a totally different perspective. I mean, next 100 years for neuroscience is going to be one of the most brilliant time, because where all these things will be tried out in some form, and already things are being tried out in small animals and kind of in the rodents and all these things. These are being tried out. When this will come to human is a different question. I mean, it's just a matter of time. It is going to happen. It's just a matter of time when it is going to happen. But currently, what are the possibilities for persons who are suffering from Parkinson disease? So, for those who are suffering from Parkinson disease, one of the therapies is called L-DOPA, L-DOPA therapy. What is L-DOPA therapy? So, this is basically, so I told you that it is the dopaminergic neuron which are dying. So, dopaminergic neurons secretes dopamine, which is the neurotransmitter. So, in the lab there is an analog of dopamine which has been synthesized called L-DOPA, and this L-DOPA is put in the spinal cord in a slow releasing manner. Okay. So, that is the L-DOPA therapy, where L-DOPA is being injected into the system. So, that is the only possible therapy currently existing for the Parkinson's disease patient. There is no other therapy currently which is successful. So, while summarizing what all we have covered, so we started with epilepsy, and we talked about the role of excitatory neurotransmitter glutamate, and all of a sudden and uncontrolled activity of glutamatergic neurons present in the hippocampus which leads to hyperactivity of the hippocampus, and thereby leading to a temporary uncontrolled non-coordinated fate of the system. One. Second, we talked about the Parkinson disease, and in that process we talked about the death of the neurons in the higher motor neurons of substantia nigra, and their death leading to not sending signal to the lower motor neurons, which are cholinergic in nature. And even if you look at the neurotransmitter profile, so the higher motor neurons from motor cortex and substantia nigra, they are communicating with the lower motor neuron with dopamine, whereas the lower motor neurons, which are sitting on the ventral horn or the ventral root or the descending root of the spinal cord, are communicating with their target tissue with acetylcholine. So, there are two different neurotransmitters which are involved in this process, and except, just for your interest, except in the case of Drosophila, the small fly, where you have the nerve muscle connection, and most of the nerve to muscle neurotransmitter involved is acetylcholine, except in Drosophila, where it use glutamate, except in Drosophila, rest all of them use acetylcholine. So, there are two different neurotransmitters which are regulating the whole motor control pathway. So, now if one component of it starts dying, which is the substantia nigra or the higher motor neuron, and that leads to something like a Parkinson disease situation, and we talked about the therapy. And then we briefly talked about the regenerative medicine, where in future probably what will happen, or what we dream that mankind dreams of happening, is that there will be neural stem cells. These will be divided and differentiated to form different kind of neuron, dopaminergic, this could be cholinergic, secreting acetylcholine, this could be GABAergic or secreting GABA, or even glutamatergic. So, and those could be transplanted for, say, Alzheimer's patient or patients with Parkinson's or epilepsy, or anywhere where there is, and same holds true for situations in other like in the cardiac myocyte or cardiac damage and all these things. So, what we will do in our next class is we will talk about another disease, which is amyotrophic lateral sclerosis and spinal cord injury, because then we will be talking about the diseases of the lower motor neurons and how they affect us. Okay. Thanks a lot.